So first of all, thank you to Tal, Nama, and Laz for putting together this really fantastic symposium. I'm Ranen, currently at Stanford and UCSF, and I'm broadly interested in understanding mechanisms and outcomes of ribosome function and dysfunction in human disease. When thinking about ribosomes, we're generally primed to imagine a factory line that simply executes instructions coded in the mRNA to produce proteins. But in fact, ribosomes exist as dynamic heterogeneous subpopulations that actively regulate gene expression by preferentially translating different sets of mRNAs. Such heterogeneity is driven by hundreds of modifications of ribosomal RNA, ribosomal proteins, and tRNAs, as well as dynamic association and dissociation of translation factors, RNA binding proteins, and chaperones or other enzymes that interact with nascent chains. These not only guide translation rate and fidelity, but also targeting, folding, and half-life of newly synthesized proteins. And together, they can form tens of thousands of different ribosome supracomplexes. In a paper published earlier this year, we showed that changes in ribosome supracomplexes during RNA virus infection reflect potential targets for antiviral therapy. We detected hundreds of such changes, including eviction of canonical translation and innate immune factors, as well as recruitment of specific chaperones and other modifying enzymes. Using this proteomic approach, we found that inhibitors of various ribosome-associated factors have antiviral activity. So if you want to hear more about this work, please come see me later. But for today's talk, I'm going to tell you about ribosomes and neurodegeneration, or more specifically, Huntington's disease, a genetic disorder caused by expansion of a CAG trinucleotide repeat segment in the Huntington or HTT gene. This encodes for an aggregation-prone polyglutamine or polyQ expanded mutant protein that causes age-dependent neuronal degeneration, resulting in physical, mental, and behavioral decline. Still, whether aggregation is the main cause of neurotoxicity remains a subject of debate. Exon 1 of HTT mRNA contains the pathogenic determinant, which encodes for the homopolymeric polyQ, followed by 2 consecutive stretches of polyprolines. Due to their structure, polyproline sequences are notoriously difficult and slow to translate. We therefore wondered whether the juxtaposition of a rapidly decoded polyglutamine and a slowly decoded polyproline could cause ribosomes to collide into each other. Given that persistent collisions were recently shown to trigger stress, we speculated this could be a major cause of cell death in Huntington's disease. So first, we used a modeling approach developed by the Tuller lab to simulate ribosome occupancy on HTT exon 1 as a function of initiation rate and tRNA abundance. As expected, at the average initiation rate for eukaryotes, the model predicts a polyQ length dependent increase in the risk of ribosome collisions just upstream of the polyprolines. To confirm this prediction, we engineered a viral display platform where either wild type or mutant HTT exon 1 was cloned into the genome of poliovirus. Within about 2 hours of infection, all ribosomes are reprogrammed to rapidly translate HTT as part of the viral genome. We then labeled nascent chains with puromycin, which becomes incorporated into the C-terminus of newly synthesized proteins, and analyze their distribution by Western blot. If ribosomes are evenly distributed on the mRNA, nascent chains will also be evenly distributed. But if ribosomes collide on a specific region of the mRNA, this should yield nascent chains of a discrete size. Indeed, expression of mutant but not wild-type Huntington generated a clearly defined puromycylated band indicative of ribosome stacking and collisions. But is this true for endogenous Huntington? To address this question, we used ribosome profiling, a deep sequencing-based approach to monitor the specific positions of ribosomes on mRNAs. Ribosome profiling of mouse brains revealed a strong enrichment of ribosomes on the 5' untranslated region of Huntington in purple, as compared to the entire main coding region in gray. Strikingly, we find that these map to an upstream open reading frame, or uORF, which is conserved throughout mammals. Furthermore, treatment of cultured cells to arrest ribosomes at the initiation step confirmed that they initiate translation from both the uORF and the main ORF, and that elongating ribosomes occupy the entire length of the uORF, but not flanking regions, suggesting that ribosomes actively translate the uORF and then terminate ahead of the main ORF, thus inhibiting HTT translation. And how does the uORF affect aggregation of the mutant, mutant protein? We transfected 293 cells with plasmids encoding GFP-tagged mutant HTT, either with or without the endogenous 5' untranslated region. You can see that without this UTR, aggregates are formed in almost all cells. However, with the UTR, aggregation is significantly reduced. And this protective effect is lost by mutating the upstream AUG to AAG, suggesting that active translation of the uORF reduces expression and therefore aggregation of Huntington. But if the endogenous uORF reduces translation, it may also prevent collisions. To address this, we tested neurons isolated from transgenic mice expressing either wild-type or mutant HTT. Mutant neurons, here in brown, had higher basal ubiquitination of ribosomal proteins and more RNase-resistant polysomes, two hallmarks of chronic ribosome collisions. Proteomic analysis then confirmed the increased presence of collided ribosomes in the mutant and identified recruitment of a protein complex that helps resolve collisions, here in purple. Surprisingly, though, translation elongation factor EAF5A, which is normally a part of this complex, was depleted. But why is that? We looked at the soluble and insoluble brain proteome of wild-type and transgenic mice and found that EAF5A is sequestered in aggregates from HD mice. Consistent with the age-dependent onset of Huntington's disease, EAF5A sequestration was only observed in aged symptomatic animals. Insoluble EAF5A levels were inversely correlated with mutant, but not wild-type Huntington levels. Taken together, this suggests that EAF5A is depleted as a function of mutant HTT expression. And finally, loss of EAF5A, a critical elongation factor, is predicted to cause pervasive ribosome pausing and collisions throughout the entire translatome. Indeed, ribosome profiling analysis of transgenic mouse neurons detected increased pausing on hundreds of transcripts in the mutant cell line. And these disproportionately affect mRNAs encoding for proteasomal and ribosomal subunits, leading to the eventual collapse of protein homeostasis commonly observed in symptomatic patients. To summarize, ribosomes are dynamic modular supracomplexes whose composition changes during infectious and neurological disease. Monitoring these changes can reveal targets for therapeutic interventions. In the case of Huntington's disease, ribosome collisions seed a cycle of ribo and proteotoxicity, eventually leading to neuronal cell death, and we propose that interventions to modulate EAF5A activity should be considered as potential treatment strategies. And with that, I'll thank the organizers, my mentors, Judith Friedman and Raul Andino, and the other people who contributed to this work. And thank you to all of our viewers in Israel who stayed up late to attend these talks. I will happily take any questions.
Thank you, Ranen. A beautiful talk. Our first question is Atan. You're muted.
Ranen, can I see you?
Sure, let me stop sharing. Hi.
So beautiful, really amazing work. I have two questions. First of all, did you look at EMs? Did you try EMs to see how you see the ribosomes?
You mean cryo-EM? Yeah. Oh, we are going down that route. We did not see anything with just negative staining, but we're trying to use cryo-EM on our virus display system to see if we can identify these collided complexes, ribosome complexes.
Mm-hmm. The second question is, do you think this is something that is outspread, is something more general in neurodegenerative diseases? And if it is, what is it doing? So do you think that alpha-synuclein and Parkinson's, the other toxins, what's the mechanism? Like, how are the ribosome aggregating, and how does that contribute to toxicity?
Yeah,
I think death of the neurons.
This is a great question. The entire field of ribosome collisions, the mechanisms and outcomes of ribosome collisions, is very, very new, and the link to neurodegenerative disorders has not been established before. So we suggested that this is probably an etiological factor in neurological disease that are caused by repeat expansions. So this could be in ALS and ataxias and so on. Whether this plays a role, ribosome collisions play a role in neurotoxicity and other neurodegenerative disorders, this will have to be established on a case-by-case basis. But yeah, I think this is a very interesting question.
Because we're seeing something very similar in fibromyalgia patients in their blood, in monocytes. We see the aggregation of ribosomes and we don't know what that means. And now I'm seeing what you're saying and I'm thinking maybe there's a connection. You know, it's like out of the blue and we're looking at mitochondria and now. So this is wonderful. Are you looking for a job in Israel?
Yes, I am.
Wonderful. I'm amazed by the talks, really. So beautiful. Kol hakavod.
Thank you.
Are there any other questions?
And maybe I can ask, Ranen, really very nice talk.
Thank you.
Get this stuff with this uORF there. Is there a gene product from this? Is there something translated? And if so, can this act somehow in trans, or is it strictly cis?
Yeah, that's a great question. So the uORF is a conserved 63 nucleotide, which could potentially encode for micropeptide, and we've tried looking for it in our mass spec data, both in animals and in tissue cultures. We could not identify it. So of course, this does not mean that it's not being made, but it could be made and very quickly degraded, or just because it's such a short peptide, we were unable to detect it. So we cannot exclude the possibility that there is some activity in trans here, but we were able, using different reporter constructs, we were able to show that the main regulatory role of the uORF is to attenuate translation initiation on the main ORF of Huntington.